Happy Friday, everybody, and welcome back for another week, episode 47 of Bioactive Live. And we're here again for another amazing, tantalizing conversation with Matthew Gates. He's back for round two. If you missed round one with him, highly recommend going back. I believe it's in the 30s, the episode number. So make sure you check that conversation out because we go deep into talking about powdery mildew, and kind of just dissecting that conversation for a good portion of 2 hours. So definitely check that one out. Today, we are going to be talking about identifying genetically resistant cannabis plants, how to set up your SOPs, how to actually know that you're moving forward with that mission at hand in the right direction. And there's nobody really else better to talk about this than Matthew Gates. So super stoked to have you, Matthew. Before we get into further introductions, and for anybody that's kind of joining us for the first time this week, make sure you check out some of our older episodes. Last week, we really hit the brakes on things, slowed things down, and got into the foundational conversation of organic growing. And if you're looking for like the beginner stuff, definitely check out episode 46 and go back through our catalog because we have a lot of information out there where we ramp into this thing very slowly. So we try to make this show accessible to all skill levels. This one's going to be a little bit on the higher level for us. And we want it that way because we have one of the great minds of the industry here talking with us, Matthew Gates. Before we get any further, my name is Mark from MI Beneficials. As always, we got Brian from Rubber Ducky Isopods and Kevin from OK Calyx. Pleasure to have you guys back. Can't wait to get into this conversation. Matthew, welcome back, and thank you so much for joining us.
Welcome back, Matthew.
Thanks for having me.
Absolutely.
So, yeah, we're here today to talk about how to set up some standard operating procedures, how to start kind of thinking about ways if you're a breeder or thinking about breeding, and how to identify plants that are genetically superior, right? And what we like to usually equate that to is pest and pathogen resistance. And so, without further ado, Matthew, I want to kind of turn it over to you because you got a whole presentation, really. You're one of the guys that's been thinking about this for a long time, and you've got a good presentation here for pretty much anybody thinking about getting into this, how you can do it at a higher level and not waste any time spinning your wheels. So, Matthew, let's turn it over to you, friend.
Yeah, I appreciate it. I'll just start with a small intro first. So one thing I wanted to mention, and I apologize, my voice is a little bit hoarse, but the information is good. So with regards to identifying resistance in plants, I want to start off by saying that resistance in plants is kind of a nebulous thing. It's hard to define even in agricultural context, but there's been a lot of research and a lot of basically theoretical talking about what makes a plant resistant, specifically to pests, but also different environments and how other factors kind of enter into it. I want to say that, and I'll submit to you proof that resistance is not really like a status. We often talk about things having or not having resistance, but it's a little bit more complicated than that. Sometimes it's not with regards to genetic resistance, but sometimes other factors are really important. And so over time, we've kind of figured this out. And a big part of that also is sort of the pest component too. So I have an example. We're going to go into powdery mildew because a lot of research about genetic resistance regarding powdery mildew in cannabis compared to other pests. So I have this presentation up here.
Yeah, and the other thing with powdery mildew is kind of what we explored last conversation is there really isn't like a great answer with environmental controls or beyond that on how to handle this. So I think it really starts with this conversation.
Yeah, exactly. Is the presentation live or visible?
Yes, we can see it.
Cool. So I've called this resistance persistence, understanding the plant-pest symbiosis. A little bit about myself. My name is Matthew Gates, owner, operator, founder of Sentinel Consulting. 14 years of doing IPM support, not just in cannabis, but other crops too. I'm going to go over this very quickly because I kind of want to get to this. I'm actually very excited about this topic. I think there's a lot of interesting things about it that breeders and growers can make use of. But I did recently contribute to the Cornell Cooperative Extension's Cannabis Sativa Guidebook, which I'm pretty proud of. And I have some other ideas regarding organic farming and IPM management, integrated pest management with regards to pests like insects and mites, and also even viruses in cannabis. So you can check out some of those publications if you'd like. When I talk about pests, like the last time we came online, I mentioned this thing called the Everswarm concept. But basically, it's the idea that pests are more than just themselves. They're themselves and the microbes around them and in them, just like plants are also this way. And so some examples of that relevant to cannabis would be like the beet leafhopper. It doesn't do much damage, but beet curly-top virus that does colonize cannabis and is an aggressive problem with no conventional solution is a huge problem. So because the virus can be in the leafhopper, its threat level goes up, right? But if you didn't know that, then you might not think to consider it at the threat maybe it should be considered at. There's other examples of that too, like the budworm moth. It has bacteria that literally break down the proteins, the toxins that are in BT when we use BT against budworms. So that's becoming potentially a problem in addition to other resistance factors they have in their body. So you can think of like the human and crop system as like one component or two components together. And then you can think of a pest system that acknowledges, regional differences, seasonal differences, adaptive differences, and also different capabilities in invasion. So one pest is no problem. If you know you're only going to get spider mites, you can kind of tolerate or hone your system to be very efficient because you only have to deal with one pest or maybe three pests. But if you're dealing with 4, 5, 10 different pests you might get, that makes things a lot more complicated and more difficult. And everything's relative to where you're growing.
Is there any way you can? Sorry, Matthew, not to interrupt, but is there any way you can hit like the present button on that screen so we can see it a little bit bigger without the side stuff?
Oh, I did actually.
I wonder why that looks like that. There we go. That looks a little bit better. Okay, sorry, go ahead.
Did it enlarge?
Yeah, a little bit better.
Okay. Can you see everything, or is it like off the screen at all?
We're able to see your upcoming slides on the left and your main slide. So that's I think what
That's weird. Yeah. That's because I clicked the presentation slide. Maybe I could try it again. Let's see.
If you go up to the top in PowerPoint and hit slideshow present
from current slide, try that again.
That's weird. It only shows. Okay. Either way, if you guys go full screen on this, I can see it a little bit better. And just do your best, obviously. But keep going, Matthew.
All right. So, like I said earlier, resistance is like an effect, not a status. So there's an evolutionary arms race with regards to pests and cannabis in this case. I think an important context point is that hosts generally can survive having a couple of parasites, like in nature. It's a common occurrence. It might allow them to maybe reproduce less, or it might be a tax on their resources. But generally speaking, if a parasite's not too much, they can have something of a parasite load. Not saying that people should have this in a cultivation setting, but it's a little different. Parasites, at least obligate parasites like powdery mildew, they can't survive without the host. So there's a huge amount of significance on the parasite having the ability to infect its host, because it'll just die. And so they respond to selective pressures that are kind of one-dimensional a lot faster. Sometimes faster than the development of certain kinds of treatments, whether they've been chemicals or perhaps biocontrols that are certain microbes or something like this. And we know from IPM the same thing is true with a lot of things with chemicals. Chemical resistance, some insects and other pests get they build up a resistance, right? And they're less effective for it. So if you have an integrated program that has many different kinds of targets, maybe safe chemistries, botanical extracts, microbes, you make the environment very hostile for the pest by manipulating it in some fashion. Or these kinds of procedures are going to be more effective, right? And pests also generally reproduce faster than plant hosts, so they have a sort of an evolutionary advantage there. A plant will grow for a whole season, and powdery mildew will produce like hundreds of thousands of spores, several of which, maybe several hundred of which, will reinfect the same host even, and then infect its brethren potentially. And in pathology, we have this thing we call like the pathogen pyramid. Sometimes it's been called the disease triangle, but over time we've added factors. So we have the genetic factors of the host and its mutualists. For the pathogen, we have its genetic factors and maybe things that affect it, like parasites, like a mycovirus. The environment, whether it's outdoor, greenhouse, or indoor for our case, and then the cultural controls, the procedures that people do who grow the plants. That's going to have a huge effect. And then time will intensify every point of this pyramid. Here's an example of such a pyramid. And for people who are not familiar with my presentations, I generally like to put the cited sources at the bottom in case you want to take a look at them so that you know where I'm drawing these conclusions from. So you can see on the right here a diagram of different kinds of things that will affect powdery mildews. Different powdery mildews can affect each other. They can catch parasites, some of which we use in conventional microbial controls. There are other foliar pathogens that might interfere with infection, although I'm not going to tell you to get other pathogens so that you can deal with powdery mildew. And there's symbionts that might exist. Most of this is on the phyllosphere, on and in and around the foliage. And then we also have, microbes in the rhizosphere, and specifically root microbes, maybe endophytes, that also manipulate and affect the plant's immune system, how it primes certain things and things like that. And that can also have mostly indirect effects on the powdery mildew.
Matthew, for some reason, we're stuck on the Everswarm screen.
Okay.
There, now it's shifted.
Oh, all right. Okay.
There we go.
So
okay, there we go. There's the pyramid.
Yeah.
Got it. Okay, perfect.
So I mean, I guess do I have to do it this way then?
You might have to do it just exactly the way, however you're doing it right now.
Unfortunate, but hopefully people can read. So the powdery mildew is kind of interesting, and maybe because I have these slides, I can turn it into an advantage. Here's a phylogenetic tree of the order Helotiales. It's a group of fungi, a really big one, and the powdery mildews exist inside of it. And what's interesting is that powdery mildews seem to have evolved from saprophytic ancestors. So basically, what we would often consider to be beneficial fungi that break down plants, their materials, their cellulose, that kind of thing. We know this because they share the same cellulase genes that break down cellulose in plants. They use the same channels like mycorrhizae use to communicate and to interact with the plant, the mildew locus O genes, which I think I talked about last presentation or in a different presentation recently. So that's important for plant cell communication. What powdery mildews also do is that when they infect the cell, they fuse with the plant cell membrane, which is very intimate and something that you really don't see if you don't deal with something that's like a strong mutualist, like mycorrhizae.
What do you mean
That's
by
very interesting.
Matthew? What do you mean by it fuses it? The cell membrane?
Sorry, I couldn't hear you.
What do you mean by it fuses it? The cell wall.
The cell membrane.
The cell membrane. How does it fuse it?
Yeah, I'm having a really hard time hearing you actually.
He's asking how is it actually fusing to the cell membrane? Is it actually passing into like the inside of the plant, or is it fusing to the exterior portion of it?
It's like this, actually. So when the powdery mildew infects the cell, we can see on the right diagram. If it's successful in the penetration, it creates this thing called a haustorium. Can you see my cursor?
Yep.
Okay, that's an advantage actually here. I can't do that in presentation mode, I think. So you can see this thing is called a haustorium. It penetrates the cell wall, but it actually then fuses with what's next to the cell wall, which is the cell membrane. So plant cells have a cell wall. Animal cells, for example, don't have a cell wall. That we're not rigid like that. So these two, so the mycelium, like the cells kind of intertwine and kind of fuse. I don't know the exact mechanics, to be honest.
Is it acting like is it parasitizing the plant at this point? Is it using
Oh, for sure. Yeah.
Okay.
So it dumps a bunch. So it's doing two things. We can go into it now. So it's a good question. So basically, the plant can it can sense the powdery mildew before it goes to penetrate, and it can, if it's very resistant, have certain genes, certain genetic responses that predispose it to react very strongly, that it can actually stop the penetration from happening. And if that happens, the powdery mildew sporulin usually dies outright. And then it also causes the priming of other cells too after it's like, hey, we just got maybe potentially infected, you know. But if the powdery mildew is able to physically penetrate through and then depress the immune system inside the cell, which a good 60% of its genome is kind of evolved to do really well, then you get this happening. Now the powdery mildew is transferring nutrients out of the cell, mostly amino acids and sugars and things. It is manipulating the plant cell nucleus it is creating genetic changes in its expression so that it produces more resources for the powdery mildew. And it will also depress the signaling of like jasminic acid, salicylic acid signaling. And it will also try to silence it from destroying itself. That's a really important critical feature that the plant cell wants to do and the powdery mildew does not want to do.
So you're saying the powdery mildew is actually is basically puppeteering the plant to not kill itself. Is that correct?
At a cellular level, yeah.
At a cellular level, which is incredibly interesting. So the plant's initial response is like, get it off, I want to die. And the powdery mildew basically tells it, no, don't do that. I need you to live, otherwise I'm toast.
You're pretty much spot on. Yeah, on an individual cellular to cellular level, usually by the time you see a powdery mildew-like colony, even a small amount, there's been like hundreds of these interactions already. Like the first 100, maybe 200 hours, sometimes more, of infection is basically invisible to the naked eye. So you don't even know necessarily that it's already happened, and until well after the colony has been successful. Yeah, generally.
Which is why, I mean, that's why one of the reasons PM is such an elusive piece of shit when it comes to plants is if it's basically attaching itself to the cell membrane, fusing itself to the cell membrane and puppeteering the plant, it's going to be very hard to eradicate, if not impossible. I mean, I would think, because I've never seen a visual display of powdery mildew with such an intimate relationship, like you said, where it's actually parasitizing and using the plant to feed itself, but also to protect itself. Is that correct? I mean, is that probably one of the reasons why it's so hard to eradicate this thing?
I like to say exactly that. I think you're right. Because and that's why I started off talking about his lineage. It was probably a mutualist, potentially a root symbiont or a leaf, somehow a symbiont of the plant potentially, or an associate with its refuse. And then eventually it probably learned, quote unquote, or found some way or developed some way to turn its ability to interact with the plant physiology into a benefit that was mostly in its favor. So no longer mutualistic or perhaps even sort of commensalic or neutral. Then it became good at kind of manipulating the plant cell. And I would also say that, yeah, so it's changing the genetic expression of the plant cells. It's keeping it from having what's called a hypersensitive response, which is what plant cells do. Our own cells do this too. If we get a viral infection, one of the first things your cells do is, well, I'm not going to be a virus factory. I'm going to destroy myself. And so a similar sort of thing is happening here. Powdery mildew cannot survive without its host. So the plant cells have evolved to individually destroy themselves and tell other cells, hey, get ready for it, which can allow it to be primed. So one thing that makes plants resistant is that they might have a really strong reaction, because if plants don't do this, like in nature, you have to be very careful with where you put your resources and how you react proportionally to something. If you overreact to something, that might not be good. You don't know what's going to happen the next day, if that makes sense.
Totally. So this also explains here, guys, why this plant over here got slammed with PM, but this plant that's literally touching this plant right here has got no PM, because from a basic perspective or basic point of what you said is some plants, they just have the genetic disposition to have that mutualistic hosting ability for PM. Some of them just don't have those building blocks or essential things for the virus to attach itself to. Correct?
Yeah, to a degree. Although the only thing
not the
about
virus,
it,
but the fungi.
no. Yeah. Well, it's true for viruses too, to a degree. But like, yeah. So for powdery mildew, for example, and other pests too, it's like the plant evolves and has mutations, and some of those mutations are super effective, and some of them do nothing, right? And so for powdery mildew, it's the same thing. It might have trouble for a time, but then after like millions and millions of iterations, it might not have that same problem. Maybe it learns, quote unquote, to diffuse. Somehow it maybe the effector that evolves has a little change in the molecular structure, which makes it 100 times more effective or something, as an example. So you can have resistance genes, but they can also break as well. Here's an example of that right here. I just want to say one quick thing about the powdery mildews, just quickly. If you're a myco nerd like me, then just know that it's been doing that for about 100 million years. And its genome is about 4 times the size of other ascomycetes or mold-type fungi. And it's primarily 60% for Golovinomyces sicroserum, which is lettuce powdery mildew that colonizes cannabis. A hefty 60 to 58%, these two strains, is where these things called transposable elements, which are genes that move around on the genome and have a habit of mutating, and often are associated with these sort of effectors that modulate the plant cell. Here is melon powdery mildew and melon cultivars that are resistant for a time to melon powdery mildew. And you can see that over about 100 or so years ago, from 1926 to like here, we've got different populations. They call them here races here for the powdery mildew. And then we have, like, for example, we have PMR, which probably stands for powdery mildew resistance 45. Like that is resistant to race 1, but then race 2 develops, and then race 5, and then and then not only do they develop quickly, but they also move across the earth quickly. And that was in like the 30s, the 40s, the 80s, the 90s. Now it's 2025. Movement across the earth is no problem. It's quicker than it was when all the continents were together, right? So this breaking of resistance and pedigree, as different groups like USDA had a hand in some of the powdery mildew cultivars that were resistant. Horticultural Field Station, this group, even some researchers from India and other countries, they would develop these cultivars in different lines, but then they would kind of stop being effective. Now, back then, the way that people would do this is a lot different than we do now. So we do it better nowadays. But it is important to understand that resistance does not like infallible. And actually, with powdery mildew, it breaks really quickly. So,
And
like
so
for example, in barley.
just to go back to the melons, so what you're saying is that as these powdery mildew strains were like developing and taking out crops from an economic level, there was different parties trying to develop resistant strains to get ahead of that, seeing that it just keeps chasing them down the stream. Is that correct?
That's totally correct.
So, and then like these melons, they're all basically the same exact melon, correct? Because I would assume that maybe like melon to a different watermelon to maybe, cantaloupe or just.
Oh, good question.
Is
That's a
that a
super good question. I'm actually,
is that a different
off
strain?
the top of my head, I did read through this paper. I don't remember if they're all the same like species. For example, like watermelon versus honeydew versus
Got it. Okay.
sour melon or all of it. I believe it is like watermelon.
Well,
If
and
I'm
because
correct.
I guess like the reason I asked that question is because it seems like there's like a common myth, and I honestly don't know if it's a myth, but maybe you can tell us if it's a myth or not. But like people growing cannabis outdoors and co-mingling, co-planting with, let's say, their vegetables, right? Where you have squash plants or melons would be a perfect example where you're seeing a lot of white powdery mildew on it. And some people will swear, cut those leaves, get rid of them, don't let them anywhere near your cannabis. And there's other people that will say that powdery mildew is not going to afflict cross species, right? It's not going to actually jump from the melon to cannabis. Is there any validity in the findings that you have? Or can you
Yes,
put that one to bed?
I think so. It's another unsatisfying. It depends, but it does depend. So here's the thing: in cannabis, we don't know all the different species that colonize cannabis of various pests, of various kind of pest groups. And in case of powdery mildew, as an example, we know of three main ones: Golovinomyces sicroserum, Golovinomyces ambrosiae, and Podosphera macularis, which is hot powdery mildew. The first one has over 100 host species recognized and is very commonly known to hybridize with others of its genus, like this one, Ambrosiae, which I call the immortal powdery mildew because that's what Ambrosiae means, but it doesn't really have a common name. I'm just going to use that because it's easier to say than some Latin wizard spell. And it's got about 25 host species or so that's recognized on, but its taxonomy is not well understood, which means that maybe what we call one thing as the immortal powdery mildew might be a different species, because some of these records were identified not by genes, because it was before then, but by the spores and what they look like and that kind of thing, which is not always effective. In fact, many times is not effective. So yes, sometimes it might be relevant, but unless you know the plant that's growing on, you don't necessarily know if you're right or wrong there. I have a live stream from the Future Cannabis Project where I go over powdery mildews and I go over the various hosts. This is supposed to be a condensed presentation, so you can check that out if you'd like, or just ask me. But yeah, so different species can have different capabilities. Here is an example of, no, so here's the thing I'm thinking of here. So I looked at different Cannabaceae, so different plants in the cannabis family, like hop, hackberry, and the lote tree. And they've got species in other genera that maybe they could bring into cannabis. Podosphera, like we already talked about. Pleocheta is found in hackberry, and Eryciphe, which is a really common one, on the lote tree. And you'll see here that Golovinomyces has an absolutely infernal 335 numbers of host genera in that genus of fungus, of which there are 64 species.
Right. So
You'll see that.
the answer is
Yeah,
really,
the answer is yes.
yeah,
Of
they can
behemoth,
jump.
yes. Yeah, absolutely. And also they hybridize. You see, they also hybridize quickly with other powdery mildews. I'm going to tell you. So you're just making this presentation this multiple slide thing actually worked really well in our benefit. So I applaud you. Here's an example of wheat powdery mildew. We have a lot of powdery mildew research on wheat in particular, and a lot of it is, I think, pretty sort of amenable to other powdery mildews because it seems like they have the same kind of strengths and weaknesses. So this is a complicated diagram, but I'm going to say it very succinctly. On the right, we have different kinds of wheat. On the left, we have the evolution of different kinds of powdery mildew. We got Plumeria graminis dicoxy, which is durum wheat, which has been around for about 10,000 years. We have Plumeria graminis triticale, which colonizes bread wheat and durum wheat. And that hybridized about 10,000. Oh, actually, sorry, dicoxy has been around longer. This one was a hybrid of dicoxy and a different species, and that gave us bread wheat. So already, when agriculture started around the 10,000 years ago, we started to get these hybridized powdery mildews. But it doesn't stop there, because about 50 years ago, so within many people's lifetime, we got triticale, and it colonizes triticale, depends on how you say it. But it also colonizes bread wheat and durum wheat. Now the thing here is that it's a hybrid of bread wheat, the triticale, and powdery mildew of rye, Sacchalis. And that happened 50 years ago. We can tell through genetic sequencing and other sort of other sort of analysis. And people would grow triticale because it didn't have powdery mildew, and now it does. And we're still reeling from that, basically. So yes, hybridizations and changes can be very radical, and we're still not. We still have the 10,000-year-old one to contend with, right? So it's not. I don't think it's outside the realm of possibility that cannabis specialized powdery mildew populations may develop or already exist.
So, actually, why don't you just keep going?
Are you sure?
Yes.
Okay. Well, where to start then? Where to go?
Well, I guess the burning question would be then, since powdery mildew it has 100 million, was that right? 100 million years of mutations and evolving. Is it always going to be ahead of us? Because obviously we're just a tiny little blip on the radar now that we're trying to breed with cannabis, and we're trying to develop, strains that it would be a wonderful world if you could say this is guaranteed not to get powdery mildew. But even just to be able to honestly market strains or genetics as powdery mildew resistant, I think that's the biggest thing people are thinking when they hear resistant strains, right? In my opinion, that's what I'm thinking, because everything else has an answer. Powdery mildew does not really have an answer except for nipping it in the bud with genetically superior plants. So, is it possible even for us to get ahead of it where we have the upper hand on powdery mildew, or does it evolve so quickly that we can't?
I think the answer is no, it's not ahead of us necessarily. But it is going to be ahead of us if we don't consider certain factors. Part of the reason I started the presentation this way was that I wanted to show a little bit about how pathology works, and then a little bit about powdery mildew, just to respect it a little bit.
Totally.
And then kind of explain how quickly these regions, like here's an example. I'm not going to get into this too much, but look at the colors here. There's this is wheat powdery mildew across Europe and a little bit of North Africa. Lighter colors are resistant to myclobutanil, which is Eagle 20. Blue colors are closer to blue. Darker colors are more susceptible to myclobutanil. You can see also, maybe not well because I expected this to be more fuller in size, but you see the copy number variations. If you have more of them, then there's more resistance to this compound because of this gene, CYP51. Okay. Similar things happen with this ERG24 gene. We have different groups here. If they're closer to blue, they're more resistant. If they're closer to brown, they're more susceptible. And what I think is important to note is that if people are breeding cannabis and they're not like looking for resistance genes, if they're not confirming them in some way, if they're not considering that different regions can be so different, then it does lower the like reliability of that resistance, because a plant could be resistant one season and then next year maybe not be resistant. But then as the meta, so to speak, of the populations changes, perhaps a population of plants of cannabis that was resistant before isn't as resistant now. Maybe the resistance is not as much of a problem, or the powdery mildew has mutated and changed enough, or you get a different species that is affected still. You know what I mean? So it can be very, very capricious. But I believe, yes, I believe we can, especially because nowadays people are looking at these genes. Here I have this example. This is a record and 3 different research reports that reference mildew resistance genes in cannabis. So we have PM1, which has a putative resistance. We don't know the actual mechanism of action here. We have PM2 from a research report that just came out, which is a really cool read. I have these diagrams from them, which we can go over if you want. What it does is it helps the plant do what I was describing earlier, destroy itself and induce a ROS, basically self-destruction. We have RPW8, which is resistance to powdery mildew 8. This one encases the Haustoria in proteins and other structural compounds that basically choke it out. So if it has a really good way of doing this, then it kind of doesn't matter post-penetration, if that makes sense. It has other proteins that work together with chitinases, which break down the fungal cell wall, which is, of course, not really great for the mildew. So if it can produce a lot of these, maybe it only produces some of these, or maybe so a mutation in the gene that allows it to do this much better. And then if you have like 4 or 5 of them that are like this at once, it's kind of like when you use 2 or 3 different rotations of a treatment that's going to not allow the resistance to develop, if that makes sense. But if we only kind of do it like a Bruce Lee flick, where the bad guy comes out and you punch him and he's down, and another bad guy comes out from the shadows and you get him and he's down, and not like getting jumped by like 40 people, then I think it's more effective. That's stunning, huh?
It's a heavy load of information.
Sorry. Yeah, but did it make sense? Hopefully, I can clarify anything.
Yeah, so
it's really good, like understanding how PM is operating and how it affecting the plants, and it's like our best chance is just to have healthy plants, good soil, healthy plants to help that plant fight back as best it can.
I think that's absolutely a big part of it. It doesn't matter if you have the right genes. Well, first of all, I'll say this: the nutrition doesn't matter if you don't have the right genes, anyways, because you're just going to fuel weapons that won't be effective. However, if you have the weapons but you don't have the right nutrition, then it's also not going to be very effective. And if you have the weapons and good nutrition, and you have other microbes, that you might inoculate, that because you have research or evidence that somehow backs it up, that specific microbes or a cavalcade of various microbes together has this really strong effect on powdery mildew, either in the foliage or in the soil. That's integrated pest management. That's you using, all the tools in the toolbox at once to disrupt and degrade as many aspects of the powdery mildew physiology, which will also make it incapable of forming the colony in the first place, then making all those mutations that it can then iterate on. So yeah, I would agree with that.
So, like you said, it's all kind of props itself up in that pyramid. In another pyramid, I think you can do everything, including adding. I would assume you're talking about stuff like Bacillus subtilis, maybe Trichoderma, probably stuff that has chitinase or can help the plant produce chitinase, salicylic acid, maybe those types of things. You can have those. You can have your environmentals correct. You can have all this other stuff correct, but which is why we're here talking about how you can develop genetics that are resistant to this thing, because there's only so much you can do. And I could tell in the chat, it frustrates people that there's not just a quick answer to this thing. Is it environmentals that causes this thing? Is it, where's it coming from? What did I do wrong? And I think it goes beyond that. And that's why we're here tonight with a science-backed person who you can tell is very thoughtful in the way that he presents this information, because I think, maybe even almost to, it's not a fault at all, but you will not commit if there is not scientific backing. And we got that from you on the last episode, which I think we should all really appreciate that. Because there's a lot of people out there selling snake oil to us. Someone on Reddit told me that I just need a dehu, and I'll never have to deal with powdery mildew again. And guys, if you're listening here, and if you're here listening, you're on the next level, because this conversation isn't happening a lot.
The way Matthew talks about PM is not how we talk about PM. We just kind of talk about the way we've heard things, and he is getting down into this is how it actually works, which I love.
I appreciate that, and I also want to say that it's not all doom and gloom. I want to start off with the context, but you ask a really great questions when I get into the details. It can kind of feel a little bit hopeless, but I think what you said, okay, Calix here, that it's if you add the nutrition, if you've got the genes, and also the microbes and other things too, then it's actually not nearly as dour. And the problem also, I think, is that breeding, has changed a lot too, and some of the standards, from an ag perspective. Let me just put it this way, if you, in my experience, people who develop resistant plants, in other crops, first of all, they have a huge pedigree, and they're known to have worked in the past. Okay. And they usually have researchers who are publishing research. Now, I'm not saying breeders have to do this necessarily in cannabis, but oftentimes they have all these resources and stuff, right? And if they were to say that a plant was resistant to the strain of the day of whatever pathogen, and they don't talk, the professionals, they talk about limitations because they know they exist. And and how do you identify resistant plants? Well, here's one. If you're talking to the source and they say that, yeah, it's resistant to all pests everywhere, don't worry about it. The plant is just impervious. Maybe ask some questions. Okay, because because in the real world that I'm familiar with, if that plant doesn't work and somebody bought millions of seeds or whatever, and it was worse than normal or whatever, then they're not going to buy from you again, and they're going to tell their friends. You know what I mean? And you probably will lose out to your competitors who maybe are doing those things. But here's where I turn back around on that very statement, because there are examples that I've even encountered where people have, I'm sure I think you guys even mentioned it too. Have you ever had it happen? I'm sure chat will agree, where you have cannabis plants that have no powdery mildew surrounded by plants that have tons of powdery mildew.
Oh yeah, absolutely. Yeah.
Oh, yeah.
I mean, this summer, perfect example.
Yeah, I always have powdery mildew on my zinnias, and the zinnias grow and touch the cannabis. But there's so far there's not been an issue with PM on the cannabis, although they are right together.
Well, and even
it could
further,
be that's not the right species, but in the other case where the other cannabis does have powdery mildew, I would look there. But you can't be sure that just because the plant expresses it here, that a different strain might not be different. Or, as we can see in this slide here, sometimes the progeny they're variably resistant. So you can take two resistant ones And if you're not looking at this, if you're not testing out and breeding out the progeny and then challenging the progeny under controlled conditions, then you don't really know. And I think that's where it gets people sometimes. It does actually take kind of a lot of work to be sure, to be really sure. And it's not to like take the wind out of anyone's sails. But nowadays, it's getting easier than ever to like genetically test their plants, to be honest. So I look forward to a day where growers can even like check it out and see like, hey, actually it has a new gene that's not on the record. Maybe it's a new one. That's actually crazy to me. I think that's actually really invigorating.
So when you actually like, is there a way to put a room full of plants to the test with a strain of PM, right? Like how does the rubber actually meet the road here? Because like, let's say you have a room full of plants, but like we talked about, if your environmentals are perfect and on point, if your soil is healthy and you're doing all the things that you can do on that side of the pillar, isn't there a chance? And I mean, there's a lot of growers probably in this chat and just in general that have, maybe never even experienced PM because they're in a perfectly conditioned environment in a factory setting with all the best equipment and everything else. Is there a way to actually push that catalyst so that people can actually run that as a test?
Salient point. Yes. So like, I mean, I wouldn't recommend growers do this necessarily, but if you're a breeder,
Right.
at a very like at a very sort of or maybe you do both, who knows, but at a basic level, sometimes people will not necessarily worry about, because going back to that pathology pyramid that we looked at before, um you have all these factors, so you'd want to control for these sorts of things. That's why people do it in labs and that kind of a thing. But in like a field environment where you don't have those resources, and you accept that things might not be perfect in your assessment or in your ability to control all variables, as Popper would say, you can never know all the variables. Then what you can do is you can just take a plant that does have powdery mildew, a cannabis plant, and it's got powdery mildew because it has the symptoms, and it's cannabis plant, not a different plant. And then you challenge it. You would just take those spores, those colonies, and you would literally touch the plant with those spores as like a very basic sort of thing. Because that way you could be assured that the transmission happened. It takes out variables like that. One thing it doesn't control for is spore load. So like in pathology, if a plant gets one spore, maybe that spore isn't even that effective and maybe just dies because it was a dud. Then you wouldn't say suddenly, oh, must be powdery mildew resistant. Usually in a scientific, an empirical sort of background, when you're doing that kind of research, you do control for spore load. So you'd have like, 10 to the power of 3, 10 to the power of 5, 10 to the power of 6. And usually as you increase the load on like a certain part of the leaf, then the infection is more pronounced. So you can tell that where the limitation might even be. And if you're talking about 10 to the power of 7 spores loaded onto like 1 millimeter of leaf, that is, just to be very clear, super unrealistic in field conditions. But you do it just to see how the plant responds.
Right.
And so in a field sort of way, you would do what I described earlier, and then see if the powdery mildew grows. If it doesn't grow, that's really interesting. You would expect it to grow if you had susceptibility. Now, if you're also, spraying like certain compounds, or if you're spraying certain, microbial solutions, obviously, that might affect things. So that's something to consider too. Especially if you're growing out, you might have like a really ebullient, microbiota in the soil, or you might have the plants primed in other sorts of ways. I want to give one example of that out here. This is Ralph Hukelhoven, and I do recommend that people, these people are just on Twitter. Okay, if you want to just talk to them and ask them a question, you can do that. they really like to talk about their work. But here's an example. Get this. The slide's called Plants, Biopriming Plants, Behavioral Resistance, for those who are only listening. We have a mock infection of barley, and we have barley infected plants with powdery mildew. And after day 3, all of these different volatiles were highly expressed. That's what the blue here is. The higher the blue, the higher it is to expression. Linalool, heptane, boronal acetate, heptane 2,2,2,4,4,6, pentamethyl. All these other things. Nonane, limonane, camphor, dodecane, tetradecane, right? Beta-caryophyllene wasn't really expressed more. In fact, it was expressed maybe less. These volatiles move to other barley and prime them for powdery mildew. So they communicated to other plants, basically, hey, there's powdery mildew. Maybe it's not that explicit, but what the powdery mildews did, what the barley did was prime themselves, and then the powdery mildew didn't infect as easily. And it seemed like methyl salicylate was a large, that's what the MESA here is. That was considered like really important. And we also know that it's a very important signal for a lot of other interactions in plants. So that can be a way that plants might be resistant that is not genetic in the same room. And that's why controlling for these things is important. It's also something we can manipulate.
My question is, how are they priming other plants? What does that mean? How are they getting other plants to produce these things or prime them, as you say?
Is it companion?
Yeah,
planting?
so the powdery mildew colonizes in this case, barley. But like I asked in this Twitter thread, do you think this would work in other plants? Is this do you feel like this is a generalized reaction, speculatively speaking? And he would say yes. It seems likely and could be a general mechanism. So what's happening is that the barley gets powdery mildew. It has an immune reaction and expresses all these volatiles. Perhaps, because part of them is to signal
through
to other
root
parts of the plant.
exudates, like through a root exudate, basically.
No, like
internally.
Like volatiles. Externally, yeah.
Like in
over the air
or whatever, yeah.
Yeah, so these volatiles are expressed in the plant probably too, and then they get out of the plant as well. And then they move around the air, and then other barley plants detect the volatiles on the air. And then those plants then have their own immune response reaction, kind of as if they were being invaded by not necessarily powdery mildew, but perhaps a fungal pathogen. It could be a generalized response. Because methyl salicylate is a common immune signaler, you could perhaps even just apply methyl salicylate or another compound and have a similar effect. And perhaps even in other pests, there are cases where you might be able to get a really strong effect. And there are reports in research where in other plants, if you prime a plant with powdery mildew against like fungi or powdery mildew in general, in specific, and then you expose them to the spores are way less effective. Because here's the thing about powdery mildew: it is assumed, quote unquote, the sort of situation it's going to be in is that it's going to be accessing a naive host, a host that has not currently already developed or has had powdery mildew or another fungus attack it. But if it's already going through an infection, or it already did in the past and survived, then those cells are going to be primed for hours, for days, sometimes for weeks, but not usually for the whole life of the plant, because immune system responses are expensive. So the plant has to have a proportional response. But plants have developed ways to detect these signals from other plants, and then it's beneficial for them to activate these immune responses, because methyl salicylate is effective at this. It is a signaling compound in many plants. It's a shared response.
So just hear me out, and I think maybe this is what you're driving at too, but it sounds like there could be a scenario where there's companion planting. Maybe not, and probably you might not even be able to comment on this, but if you were to companion plant with something like barley, and you had barley and you had a strain of powdery mildew, because we kind of know, or from what you're saying is we kind of know there's like 3 strains that afflict cannabis plants, right? 3 specific strains
that we know of.
that we know of, right? So this would be definitely a risky maneuver to try, but listen, like we're desperate. I feel like breeders, if you can actually get your head wrapped around this and get a system in place where you can actually start to spend time doing the right things, this might actually be gold for you. But is it possible to have like a companion planting of like barley with cannabis in the same area where the barley is sending this methylsilate out into the air? And is that going to be received by cannabis the exact same way that another barley plant would? Or is that not so clear?
I think that's plausible, because methyl salicylate in particular is like a very common what we call conserved signal compound. That's actually why I asked that question. Now, you might not even need to do that if you don't want. You could simply, well, there's 2 things this makes me think of. One, companion plants, like you said. You might have a plant that, here we go, hosts a powdery mildew species that you know doesn't affect cannabis and you're pretty sure won't affect cannabis. Let's say maybe there's a tree in your backyard that gets powdery mildew all the time. Who knows? So then the plant, maybe the tree or the plant or whatever, it reliably gets powdery mildew and expresses volatiles, and maybe those volatiles, I don't know how you would confirm this, but perhaps it would prime the plant. Yeah, that's plausible. Another thing that could happen is that you just apply the methyl salicylate at a certain sort of rate. You have to be very careful with these kind of signaling compounds, kind of like hormones. Too much is maybe going to give you a very wonky, problematic effect. And it's easy to maybe do too much or too little. But assuming you could do that, you could prime the plant. Like, let's say, and here's where IPM comes in, and having your crop scouting good and your records and everything. If you know that, let's say you're an indoor grower, you tend to get powdery mildew in winter. You don't know why, but it's almost like clockwork. Week 30, whatever, week 35. So week 33, you start hitting them with this volatile, or maybe a different volatile, whatever it might be. And basically for 2 weeks, you kind of tell the plant to up your defenses. And then week 35 rolls around and you don't get powdery mildew.
So that's amazing. So these volatiles are actually available. Like do you have to be like at a scientific level to be able to use these things? Or is this something that like a breeder in the basement could actually start playing with getting their hands on some methyl salicylate?
Farmers, agrarians, growers are very clever people. So I think there are ways to do this, and I think there are ways that people could experiment with this responsibly. I think you can get access to these things. A lot of these compounds, yeah, pretty available. I think you would have to be careful about concentration a lot. But like in kind of like a crude sort of way, yeah, I think that there are ways that this could be effective. There are products out there. This kind of research is done because people have tried to do this in other ways and have been somewhat successful sometimes and less successful in other times. Even myself, from a professional standpoint, I've told people like, hey, this is a really cool idea, but it's kind of like experimental, or it's very hard to confirm in the field if it's working or not, so it's like people don't want to try it out, which I think is valid. But yeah, if you wanted to experiment in this way, I think that that is a plausible thing to draw from this information.
The other fascinating thing, Matthew, that I've just thought of here is that it's so there is a risk, right? We were talking about other plants that have powdery mildew in the garden being close to cannabis. But I would be willing to say that it's a minimal risk for there to be, because there's only 100 of the species that afflict cannabis. Of those 3, that first one had about 100 other known hosts, right? Which to me is relatively small, and I would assume I'm gonna make some stupid assumptions here because I'm not a science guy, but I would
Powdery
assume that
mildews are often specialists.
I would
I'll just
assume
say this first. Yeah, powdery mildew are specialists. Most of them are not generalists. So
right.
so
So
actually powdery mildew having 100 plus host species is kind of amazing. Kind of like here, you can see a lot of them are like 1, 6, 1 for this genus, 3. Somehow, oh, this is an outgroup. But only a few of them have like lots of hosts.
right.
Yeah, you're right. Yeah.
So I guess what I was trying to say is so your risk of those plants, those other plants, those different species of plants that have powdery mildew, of actually giving your cannabis plants powdery mildew is pretty low, relatively low, but
Plausibly low.
plausibly low.
Nominally low.
But the benefit, the potential benefit, is that they're releasing this methyl or these volatiles, right? Not just the one, but all of them into the atmosphere. So they could actually be helping your cannabis plants from resisting powdery mildew in some way, shape, or form. So just food for thought. I mean, I'm not saying
Yes.
that's like a case closed. Keep them, and don't even consider chopping down those plants with PM. But just another consideration.
That's why I
And
brought this information up. I love that you made that point because that's why I wanted to include it, because I think there's these really cool ways that people can use companion plants in this sort of um, unorthodox fashion because they understand, evolution, physiology, ecology. People who are trying to be very sort of eco-sensitive and intelligent in using these factors. If you know this stuff, then you can draw these conclusions. I think that's really great lateral thinking. And for example, if you were to look at this, you might decide that, well, maybe I'll not get the hosts that get Golovinomyces. Maybe I'll get something that is infected by microdium. Maybe I go look that up, or I hire a person who knows this, or something else like maybe not Pleocata, or maybe not Golovinomyces, or maybe not Potosphaera, and maybe not Ericyphe because they're in other sister groups to cannabis, right? So you would choose things that are not in these groups. Fantastic conclusion.
that just blows my mind because we talk a lot in living soil, big volumes. Steve Cantwell, I saw he had a question for you on Instagram.
Yeah.
I think you saw it. But there's a lot of guys. I bring up Steve Cantwell because he's doing this on a huge commercial level with Green Life Productions, and we do companion planting for different reasons. For like natural mulching, for root exudates, for diversity in the soil and stuff like that. But no, I don't think I've ever heard anybody discuss the actual volatiles that are coming off of that. So considering maybe there's a way to steer our decisions with which companion plants we're putting in our beds indoors, because outdoors is there's different reasons for doing it outdoors. Indoors, in a kind of a closed air loop, certainly more intimate setting, there might be something more to picking your companion plants with what we're talking about here, and the volatiles that they're going to put off into the air itself. We really haven't talked a lot about that in the cover crop discussion. So I think this is really groundbreaking stuff here.
It's kind of ephemeral, and as a professional, it can be hard to it can be hard for me to say yes, this will definitely work, or no, this will definitely not be effective and worth your time. So most people will pass on it, and it's understandable. But as we learn more about this information, I think it becomes a lot more tenable as a prospect, especially if we observe some basic safeties like, like I said earlier, not getting a genus of powdery mildew that's associated with cannabis. Sorry, a host that sources that powdery mildew. Yeah, and I also think that in an indoor enclosed space, you can then manipulate the psychrometrics differently, like humidity and other sorts of things. And perhaps in this day and age, they're looking at a lot of biosensor work. and biotech for agriculture. And so it might be very much more possible to get a reading in much the same way that people do readings for CO2 or oxygen levels and that kind of a thing. You could perhaps get readings of these sensors that are reliable enough to know. Maybe you'd even be able to tell that your plant has powdery mildew before you can visually see it, just as an example. So I think that stuff is really fascinating. And also in this companion planting, perspective, here's another example. Maybe you could host microbes on those companion plants that are also effective at, supercharging this reaction that you're also counting on as a volatile for your plant. Also, the effect on your own cannabis specifically, and then also potentially other microbes that would disrupt it in the phylosphere as well. So you kind of get like a 1-2-3 punch that way. If you play your cards correctly and actually know with great detail, what the interaction is going to be. And I think that's where the key is for a lot of this really cool ephemeral stuff.
That's great information.
Would you show us the 3 strains or genre of PM that affect cannabis again, please?
Yes. Lettuce, immortal, and hop that we know of so far. There could, and I think are likely to be, others.
So
sorry,
do you?
Are there plants? I'm sorry, Mark. Are there plants that PM just will not affect? Like they just cannot get through it, or doesn't ever mess with it?
Oh, I don't know. I think that, well, my opinion on that is that, and that's actually why I included this section here, the early hosts of powdery mildews, as we know them today, as parasites, they evolved in northern polar forests, kind of up here above this green line. And over time, they moved down 100 million years ago or so. And so you got birches, beeches, cashew, maple families, the figales and the sapindales. But then they colonized the asters. So about 58 million years ago, as they were evolving around here and moving up into Africa, what would become Africa and North America and Eurasia. And what's important about that is that here's a primitive aster, sort of a sunflower family. And here's what you would normally see nowadays as an aster, kind of a sunflower-looking thing with a composite amount of little florets in the middle, which become seeds. And basically, they're the most speciose group of flowering plants. They're about 10% of land plants, I believe, or flowering plants. So what I'm trying to say is that powdery mildews got really successful. Some of the other first powdery mildews, or the most basal powdery mildews we know, are also common on oaks. So to answer your question, they're pretty gregarious, and I don't know if there are plants that just don't get powdery mildew. Perhaps there are. I imagine that there are probably plants that are not super vascular, by which I mean either non-vascular plants or something that comes to my mind are like stone plants or like cacti. I don't think cacti get powdery mildew. Some of these sort of succulents maybe don't get powdery mildew as much, to my knowledge.
Yeah, there you go. That's a good point.
Yeah. So if the cannabis doesn't work out, maybe mescaline next time.
San Pedro, baby.
Yeah, there you go. If powdery mildew is just really not working out for you, you can try something else.
So we've talked about a couple really kind of interesting ideas, and so does it go further than that? Like, what else would you recommend for somebody who's trying to really, like a breeder, right? So bringing in a plant that has powdery mildew, seeing how it affects things, culling your plants, culling your picking your males and your females based off of the reaction or lack of reaction that they have with that introduction. But then it sounds like just as important is running that same test out with the next line of genetics, or with those babies that come off of those plants, to keep that moving down the line. How far do you think that process needs to go to really be comfortable in, marketing? Because I mean, like, so to market something as resistant, it does not mean that it is not going to get it. It means that you're going to have a better chance of not getting it, right?
Or not getting it to the same severity. Yes.
To the same severity.
That's typically what people are expecting. Yeah. Yeah.
So how, in your opinion, like, how far out would you run this to be able to comfortably say, okay, we've got something special here?
I don't think I'm the appropriate person to ask, but I can give you a speculative idea. I feel because I'm not a breeder and I don't know the exact processes, but I do know that in a basic sort of way, if you were to ask me that 10 years ago or 15 years ago, then the answer might be several years. That's why I said that resistance can break really quickly, because while you're fielding the first one, the powdery mildews are kind of adapting to it, and or rather, it should also be said that with selection pressures, the powdery mildew might already exist out there, and you're just culling the ones that don't work, and the only ones that do work you see, if that makes sense. Kind of like with insects and pesticides, the resistant insect already existed, and then you were selecting, or at least to a degree, and you're kind of selecting for progeny that are more akin to that. So I don't know, but nowadays I think that the ability to what we call pyramid these genes, these resistance genes, together through breeding, and also through marker-assisted breeding, where you can look at the genes. That's what they did in this research report, by the way. Kind of, they were able to look at the plants. They were able to look at all the progeny, and they were able to go like, doesn't have it, doesn't have it, doesn't have it, won't be good, won't be good, won't be good. Got rid of all those, grew out the rest of them, and you obviously, even if it has the gene, it might not express it to the same level, or there could be other sort of variables that are comorbid. So you probably have to do it still, a few years to be, sure to a certain degree. I think that it really depends on the resources you have available, though. What we're talking about here is like, biotech stuff. But more and more of those technologies are becoming democratized.
So how about this, just real quick as a follow-up to that, and we have about 14, 15 questions that people have for you in the hopper. But my follow-up to that would be, I know what you're going to say because you're a science guy and you like a controlled environment. But from a
that's not true. Hey, no, I don't appreciate that characterization at all. Wait,
Shots fired. Shots fired.
Whoa.
I know you're just a
Hey, I want you to know this. That you're right. I mean, there's advantages to this, but in my world, when I grow plants for just my own pleasure, I love wild overgrowth, actually, probably to a detrimental degree. I just think it looks cool. So maybe that's something repressed by being an IPM specialist. But
yeah, so this is a good question for you then, because what I'm wondering is it from a practicality standpoint, right? Maybe we're not trying to find the answer to the exact question, but from a practicality breeding standpoint, do you think it's more effective to find a resistant genetic line by growing it outside as opposed to inside? Because outside you're going to be exposed to a lot of these things. You maybe even exposed to all three strains that we know afflict cannabis. Is it, if you're afforded the ability to grow in like a greenhouse, or just directly outside, kind of like Masonic, who lives in Compton, it's got perfect weather all year round, he can actually throw these plants outside if he needs to. Is it better to run this process outdoor or indoor for a stronger genetic resistance?
To to be sure that you're accounting for variables, it's better to do it probably indoor, but then you test it in a field condition afterwards, I would say. If you were trying to be like, if you were trying to like challenge the plants, you also might consider like kind of trying to the best of your degree, sort of sterilizing the foliage in some way, right? Because you might have microbes. I mean, it depends on what you're expecting and what you have control over and what you can verify, right? But let's assume that you're like, hey, I actually, use various microbial sprays that, can affect powdery mildews allegedly. So I'm going to like stop doing that. Maybe I would apply something to sterilize the leaves or whatever, or I would just stop using it for a while. Then I would try to challenge it just to be sure that we're testing like the genetic reaction rather than like the commingling of various factors. And then what you do is you take it out in the field, um, but you have to make sure. But you also want to like check your progeny too, because you're going to expect that some of your progeny just aren't going to be the top of the top, probably. Um, so that's where like that's the typical breeding problem, right? Is like if you have all this wizardry and technology at your disposal, then you can like do marker assisted breeding. If you don't have that, then you need like lots of repetitions and probably like lots of individuals to test. So it takes longer that way as well.
So it sounds like really just doing both, like really just running the
Yeah.
gambit. Don't do one and just expect it to translate to the other. Testing it across the board is going to be your best bet, and also try and like that was kind of why I prefaced with I kind of knew what your answer was going to be, but like being
Yeah.
able to establish the control is important for getting a solid answer. So
It's for being sure,
yep.
because it's possible that you selected everything right, maybe, but like you won't know until those powdery mildews come back, and it might be an unhappy turn of events, So like it's just it's a reliability thing, but there are people out there who I've encountered where they've had what we described earlier. One plant, no powdery mildew. Or one group of plants, all the other plants around it, tons of powdery mildew. So I would look there too. Something's happening, probably.
Yep. All right, so we're gonna jump into some community questions.
Let's do it.
And I want to start with this one that came from Steve from Green Life Productions. It's on Instagram. And I think we kind of answered it in this discussion. So, Steve, if you do get a chance to go back, if you're not satisfied with this answer, definitely go back and watch this episode. But the question is, so we know certain environmental conditions can favor PM, but as far as resistant from genes, I wonder what conditions turn on or off certain genes or traits from an epigenetic perspective. And would a healthy soil biome help better prime the plant for resistant versus synthetic hydro systems? Do you understand the question?
I think so.
So he's saying we know that the environmental conditions can favor PM for growing, which we can probably
True.
agree with. Is the soil biome it goes back to the pyramid, right? It goes back to the
Yes.
genetic ability of the plant, the genetic superiority of the plant. It goes with the environmental conditions, and it goes with what you have going on a microbiologic level in the soil and on the plant itself. Is that correct?
Yeah. So like epigenetically, that would be so that's a thing that happens for the expression of genes. I'm actually not very familiar with the epigenetic dynamics of cannabis in particular, and not a lot of research into it, and also with regards to like powder mildews. But um, like during the infection, there's something to be said of the plant's ability to resist sort of being compromised by the powdery mildew and all the little effectors that it secretes out during its colonization. So things that will affect that absolutely would be not only the intrinsic genes of the plant and the how the chromatin unravels, and methylation is often a factor in turning off and on genes, but it's kind of hard to track. Not sure how we'd be able to verify that in field. But soils in the, like I said earlier, in fact, I put this in before he even asked that question, but like these things, other pathogens, other symbionts, parasites of powdery mildew in particular, we have examples of. Definitely rhizosphere microbiota, like what okay, Calix was talking about, microbes in the soil. It doesn't have to be priming the plant, because the fungus being there, for those who don't know, even having symbionts that are fungi, the immune system regulates these symbioses in a very sophisticated way. And so just having mycorrhizae on, in, around the plant roots, it causes the immune system to kind of sort of look at fungi a little bit more seriously. I'm really grossly simplifying it, but that's what happens. And so just the mycorrhizae being there can help out the plant, prime it against fungi and generally, and perhaps even powdery mildew. So that's definitely a factor in the soil microbiome. Now, there's also going to be microbes. Again, hard to verify which ones and that kind of a thing. But there's definitely microbes that maybe are detrimental or maybe help the powdery mildew in direct or indirect ways. And that would be something we'd want to research to find out more about. But it can be very like, needle in a haystack kind of thing.
All right, Brian, let's jump into some community questions. And guys, we really appreciate everybody for hanging with us. And this is like a really jam-packed episode here, full of information. And if you guys are liking the info and you are enjoying the show, don't forget to hit the subscribe and the like button. It really helps us so that we can get this out to more people. It's a free show. We support it with just the funds that we get out of our own little small businesses. And because we really enjoy this conversation, it's because cannabis has been stigmatized for so long. This conversation is just now being able to be possible, and young folks like Matthew Gates that are bringing this information to the forefront, really pioneers, and helping people that are breeding to be pioneers in their own regard. So thank you guys for sticking with us. And Brian, let's turn it over to some community questions.
Yeah, absolutely. I think we have a newer listener. This Jorge was asking some questions at the beginning. I got one plant way bigger than the rest. Should I crop it? That's for more of like the panel at first, and then the rest are for Mr. Gates.
That would boil down to your own personal restraints, right? Like you have a size or grow space constraint. That would be up to you. If it's going to be blocking out light, stuff like that, those are the considerations that you would need to take into effect. If it's out of control, stuff like that. So that's a pretty loaded question. But I would say that you got to take that with all the other things that you have going on in your grow. If it's going to be pushing the light, obviously you want to chop it. If it's a male, you want to chop it. I mean, or if you're trying to breed, maybe you don't want to chop it. Yeah, that's how I would answer that one.
Kevin, you want to add
I
anything?
would just say that since we talked about genes, just real quickly, some genes are on the X chromosome only, and some are only on the Y chromosome. And I've seen some research that maybe sort of intimates that you don't really need the Y chromosome for the things you care about. But I don't know how true that is. As a Y chromosome haver myself, I don't really like that tone of voice. So I don't know. I don't know. Let's do more research before we get rid of all the males, huh?
Especially in the earlier days, you would hear this a lot. But isn't PM just humidity issue? Why not just get a de-humidifier? Even Mark kind of mentioned it because it's all over the place on the internet.
Yeah, so I've heard that a lot of times, and I think even last episode I said the same thing. And like people will say something like, I've had clients where they're like, "So I got all this good dehu and all this stuff, but I still have powdery mildew. Isn't it kind of like this? Just a question of humidity?" Well, no, it's a question of having the spores, right? So the spores are hitting your plants, and It's not spontaneous generation. So you want to keep the spores out potentially if you're in an indoor area. So it's a biosecurity question, really. How is the powdery mildew getting in your area? And actually, powdery mildew, unlike a lot of fungi, does really good in dry conditions, actually, because the spore can hold a lot of water in itself more than other fungi can. So dry conditions work out too. Sorry.
All right. Next question from Kevin's Instagram. Does labs work to eliminate PM?
I think there are some labs that have been shown to do that. I think also, I don't remember the mechanism.
He's referring specifically to lactobacillus, like lactic acid bacteria, right?
Yeah, which could be lactobacillus and other ones too, right?
I
Yeah.
think it's just lactobacillus, isn't it?
I don't actually remember. Is it just lactobacillus?
It's just lactobacillus. There's another. If you go with like effective microorganisms, there's more than just one strain in there. But labs is purely just lactobacillus.
Okay, cool. I appreciate that confirmation for me. So in that case, I mean, eliminate. That's a crucial word here, because I think it might even be most effective preventively, by disrupting the environment. Like I said, making it really hostile, because the powdery mildew spore comes in, and other things that affect powdery mildew that have been used in the past were, or nowadays too, is like things that affect the pH of the surface of the leaf structure, which acids will do.
Awesome. Yeah. The other was I think that the other thing I've heard is spraying milk on plants kills powdery mildew. Have you heard this?
I have heard, and people have done tests to see this, and sometimes it has been very effective. But I think there are, and I forget the mechanism of action. It might be some of the proteins inside the milk that are enzymes that break it down or something. But I think you get other sort of second order, third order effects that would make it difficult to use.
I would say, to be clear, I think it's the raw milk that actually makes this. So if it's been pasteurized, if you go and buy like the really cheap milk, I guess there's still debate out there, but you would want to get.
I think when they tested it, they did. I don't think they used raw milk when they tested. Are these the things that I remember? But perhaps to be even more effective, that's a great point. Yeah, a lot of those enzymes are heat sensitive.
Exactly. There's one more question with PM about can you spray PM with the peroxides?
Yeah, you could. They are not a big fan of oxides in general, especially when the plants are generating them in the plant cells, right? But yeah, you could do that. I think that if you are going to use like a peroxide or other sort of similar kind of sort of antimicrobial, I try to rotate other things too, just to like really mess it up and increase your success. And of course, dealing with a large colony is different than dealing with a small amount. So try to get it in its earliest iteration.
Well said. And then Rob over here, I've personally gone through this, and especially the early days with just like this little tent where the plants got PM. So I just wanted to highlight this to people that are newer to cannabis farming. I think there's a lot of just blind, enthusiasm. So if you go away for the weekend, you can kind of come back. That's why they call this, especially at the beginning, the golden handcuffs, because you can't necessarily always just run away for the weekend. So just had to kill my whole flower room because of PM. I have a de-humidifier and kept the humidity way down. I did a big dry back and one plant exploded. Then before I knew it, over half the plants had it. So just wanted to highlight this kind of stuff. This is why you want to be extremely proactive, especially in the early stages of this, because you don't even know what's kind of potentially coming your way. And you're going to think, especially in veg, oh man, this is easy. Because a lot of us, veg is kind of easy, to be honest. And especially in a brand new setup, there's nothing really coming to attack it yet. So remember to kind of keep your mind on being as proactive as possible. And if you're watching us, to be honest, on a Friday night, you're probably already a proactive person, because there's a lot of fun.
Look at you, you're doing it right now.
Yeah, exactly.
Right?
I mean, the Friday nights are fun, but at the same time, man, you got to make a living. And at some point in life,
it's not fun powdery mildew destroying all your flower. So
Yeah,
yeah.
really bad.
Especially when it could be prevented just by being maybe a little more educated on the subject. This was actually my question here. Does myclobutanol turn into cyanide when heat is applied? We had always heard this from the hippies and from some of the state representatives and stuff in the early days.
I forget actually off the top of my head if this is true. I want to say yes, and maybe it's a similar compound. It's not
Okay.
great. It's not
Yeah.
great to burn it up and inhale it.
Yeah. You definitely want to think about these things when you're
Avoid
adding
that,
heat to
yeah.
them. From OK Calix himself, what environmental conditions are known to trigger PM growth?
So I'll start off by saying something that actually powdery mildew ironically doesn't do very well with, which is actually high moisture on the leaf itself. So like if you had water, and there's other reasons you wouldn't want to do this, but if for whatever reason you had powdery mildew spores on your leaves and they like got doused with like water and like sit there, they would drown. In fact, I have a friend who's a researcher doing research for powdery mildew resistance in cannabis. And he said that he had to play this scrambling game where they would collect the spores off of the leaf using some kind of like a collection system with like a liquid. They had to run really quickly to go and isolate, analyze them. Otherwise, the spores would die. So that's kind of an interesting point. So things
And that's
that do help them, though,
contradictory. That's
it is,
contradictory
right?
to what people are spreading on the internet.
Yeah. And like other fungi, but other fungi will do great when they have lesions and like suffocated, epidermal cells. So it's like, you don't want to do it either, but it's an interesting thing to think about. Um, One thing that once a colony actually, so nutrient stress is a big one. That's a huge condition. The host genes being kind of weak or somehow inhibited somehow, like with nutrition or certain other pests being active, that can also inhibit the plant's response. Another thing that is helpful for powdery mildew, especially if the colony is already there and you got the conidiophores, the spores they're producing, it's the rapid change from dry to wet to dry. So dry, humid, dry will cause the conidiophores, the spores, to just kind of launch off. So that can maybe, to a certain degree, increase the level of colonization. As an example.
Totally, because that's that's kind of the tale we tell here in Michigan. I'm sure it's the same elsewhere. Is it's that late season. It's like the September, October where you're getting that warm day, cold night,
Yes.
high
Yes.
dew point.
That's it.
So there is validity
true.
to that. It's good to know that there's validity to that one.
Yeah.
But it's
Yeah.
such a nuanced conversation that it takes so long to get there, and I'm so glad we got there. Hell yeah.
Humidity, like powdery mildew hates rain. It destroys powdery mildew. But then the spores that still are there around after the sun comes out, then you have problems. And the powdery mildew loves that. The ones that survive.
Our next question is from Mark Nelson. What can you prep with to help stop PM like any other plants help hold it off? I think
You
he's
made a
talking
really
about more companion. Yeah. All right.
We kind of hit
I
on
really
this,
liked
Mark.
the companion plant idea. Yeah, no. Yeah. I mean, I think what we talked about just now was a really cool idea. But it's also one that I don't really have a lot of background with, but I would love to see people try to like use it, try to kind of record that, because that's really fascinating, and that's where a lot of ideas start, is with that experimentation.
Another question from Mr. Kevin himself. It's a good topic when Kevin starts firing them off. Can you give us a few store products or organic inputs or microbiology that works well on PM?
Yeah, there's various strains like you already mentioned, Bacillus subtilis. People like to use. I'm actually a huge fan of using potassium bicarbonate as a product because it disrupts the pH of the surface of the plant, and so things that are like that can be really effective. You don't have to necessarily get that specifically. What I like about it is that it's pretty neutral in the like grand scheme of things of what it affects and how it affects your plant, as far as I understand it to be. So it's kind of a harmless thing that you can apply multiple times, especially if you think you're getting to a seasonal time where you get peak powdery mildew or when it starts to start. Um, a lot of people like to use micronized sulfur as well, especially if they tend to get it in a really humid propagation room. That's pretty common. It also affects a lot of other things. So, you have to be smart about it. You might dip your cuttings or something in it, and then later on, you might want to reapply with potassium bicarbonate or another micronized sulfur spray, or maybe you do that, you let it kind of rest for a while, and then you start incorporating other microbes in the phylosphere and the rhizosphere, like some of the ones we already mentioned. And actually, I don't know for a fact, but like I said earlier, if you do use mycorrhizae or you're thinking about using it, I would recommend potentially doing that because you might get some of those cool priming effects we mentioned.
And all the other benefits that you get with using mycorrhizae as
Exactly.
well. It's just a win-win.
It stacks up.
I did see a comment in there. Someone said that they heard on a Kiss Organics, or it doesn't even matter whose podcast it was, because this is the disinformation that we're talking about or not disinformation, the misunderstanding, I guess, would be the proper way when it comes to powdery mildew. But so, they heard on a podcast that if you use ample amount of silica, you'll never get powdery mildew. We can pretty much, from the conversations we've had, we can agree that that's just not true. Can't be scientifically backed, etc. But does things like silica or chitin or the other one I've heard of is salicylic acid coming from like aloe, are these things too that you would recommend?
Yeah, if you use them appropriately, I do. There's lots of research, and in my more, sort of in-depth, if you really like the depth of this one, then you should check out that Future Cannabis Project live stream where I go even more into depth on the physiology and that stuff. But silica has been shown to be used in various ways. Not only does it prime the immune system as a signaler, it also ends up being used in the sort of cellular structure of the plant, almost like putting like metal plating into like housing so that it's a little bit more defensive in a physical sort of way during that penetration we talked about earlier. So research definitely backs up how that can be effective and helpful. Does it mean you'll not get powdery mildew? It's very contextual, but there's solid backing for that. Salicylic acid is the pathway that primes plants against a lot of times different various kind of fungi. Powdery mildew will even try to disrupt this, or even sort of increase jasmonic acid pathway signaling, because that is antithetical. So it's the opposite of SA, and so that will then disrupt SA effects, if that makes sense too. So if you use these priming, compounds in this way, yeah, you can make a really good effect.
Follow up, or not really a follow up, but is there a company that you can send cannabis material to and they check the plant's genetic resistance to PM?
Yes, there are. There are a few out there that I'm familiar with, but I don't really want to talk about it too in depth in case I don't have the right info. I don't want to give people the wrong impression online. But ask me that question another time, or I could come back later and answer in the comments or something to give some ideas. There's also places you can go for other pathology testing as well. If you wanted to check to make sure, not that you have powdery mildew, but potentially, what strain in the future, what species. That can be helpful more in the future.
This next, or one little shout out, I guess, to final out all the praise tonight for Kevin. Okay, Calyx Beans, tough as fuck. No PM on mine. So just throwing out that Kevin's doing what he needs to be doing.
And Brandon Russ
There you
just
go.
chimed in from
what up?
Bokashi Earthworks. He said medicinal genomics or genomics is an option there. So just to throw that one out there.
Thanks, Brandon.
For PM testing.
What are your thoughts on that? I run a small HEPA filter in my grow. If there's a PM event, it really helps. Is that a placebo a little bit there?
A placebo?
Well, like, is it more in your head there? Like, it might work a little bit, but I mean, it's not There's no way
Well,
to.
if you're on your intake, yeah, it will probably if it's small enough, which it should be, then yeah, you shouldn't be getting spores through that. And also remember that unlike black mold and other kinds of molds, it's not like it can grow like on the surface of a dirty wall or something like that, or some debris. It really needs a living host to grow. So you don't have that problem, fortunately.
Right. Dr. Crispy Thumbs. Any suggestions on how to treat Septoria? Is that how you said that?
Yes, yeah. Yeah, Septoria or Septoria, as it was recently changed for some species, is like powdery mildew, kind of. It's sort of a surface level fungal pathogen. Causes a different kind of damage, though, sort of like a necrosis, browning, which usually has like a purple halo around it as it causes yellowing as it spreads. And a lot of the things you would use against powdery mildew actually would work against Septoria, like the big ones, the heavy hitters like micronized sulfur and that kind of a thing.
And this is the last question from the audience, or the main ones that were there. If you thin your plants like crazy, like defoliation, will that give a better chance to hold off PM?
It's an interesting question. Because also when you do all that defoliation, I don't know how if this is quantified anywhere, but you are kind of stimulating mechanical damage that you'd associate somewhat with like a mammal or something eating the plant, which can have immune response effects. I wonder perhaps if that might be um, in play. I don't know. But that's where one thing comes to my mind. For the humidity and other sorts of things, that could be helpful potentially. But like we said earlier, powdery mildew, unless it's very extreme one way or the other, is probably still going to be good to go.
Yeah, there was a Japanese research. I think this is a long time ago, but I think they were using brine shrimp, and they were frying the brine shrimp in like hot boiling water, and then hooked up the plants to something electrical, and we're getting results from that. And it was basically like trying to simulate the same part if you were cutting down the plants and all these plants are around them. So maybe I could go back and do some digging into the internet and find that. But I just thought that was a very interesting kind of setup that I have never seen anybody ever try to speak on or replicate. And I wanted to throw that out there to you. So do you think brine shrimp, the plants were actually kind of feeling that sensation that they were in distress?
Oh, is that what they were trying to do?
Well, they were using the brine shrimp to create the problem, and then recording
What problem?
how well, simulating like when you would cut down plants, do they know that you just killed their brother or their sister? And so they were using brine shrimp to kind of simulate this. But I remember it was like in the late 70s, early 80s. I don't know if
Well, I'm
anybody
not
else
sure.
has seen that
I
documentary.
think. Well, one thing you had mentioned earlier was like with like chitinase and chitin. So like being exposed to chitin, even like from a beneficial fungus, does make the plant go, maybe I should be a little bit more, fungal conscientious with what I'm developing. So in that way, if you had, because chitin is also a polymer that's in insect carapaces and that kind of stuff. So maybe exposure to chitin in that way, which some people use as amendments, right?
Yep, absolutely. Shrimp, fish, and crab, or not fish, but crab meal, shrimp meal, stuff like that. Always good and good source of chitinase. I also love barley. Barley is a great source of chitinase, and I wonder if growing barley beyond the volatiles that it could potentially release for your plants beneficially, if there's also things happening on a root exudate level if they're used in companion planting. The only problem is barley is pretty aggressive in the way that it grows, so you'd have to be pretty tactful with the way that you manage that that commingling, if you will. So this has been an insanely insightful conversation, like way beyond what I actually what I ever thought it could have been. So anybody who came into this thing late, definitely go back and re-listen to this episode, because we hit on some points that I don't think there's a whole lot of conversation going on about this kind of stuff, and it goes far beyond just what you're finding on Reddit or what you're finding in the vacuum of the internet. It is a much more it's a deeper conversation than that. But there's a whole host of things that you can do to help yourself. And if you're from a breeding standpoint, there are actual things that you can do to cull resistant plants. So make sure you go back, watch this thing, and also go back and watch the first episode with Matthew. We had him on 3-4 months ago, so it's probably episode 30-ish, where we talk about this a little bit more on a broad level. We definitely hammer it down in this episode, and it's been such a great opportunity catching up with you again, Matthew. I can't wait to do this again soon. With that, how can people get a hold of you if people have more questions, actually need consulting from you? Because I think that's what says Zenthanol Consulting right under your name, but that's what you are with Zenthanol. How do we get a hold of you, and what exactly do you do?
What is it that you do here? That's a good question. So yeah, Zenthanol
That was
Consulting.
wait. That was the Office Space reference, right? What is it you would say you actually do here?
What exactly is it that you do here in particular, right?
I just like that.
No, you're right. Yes. So IPM support basically. So if you need help with pests, in emergencies, or better proactively, if you're trying to improve how you go about things, I think it's a really good investment. I usually tell people that the way to look at it, I think, is you can either have a certain percent of 100 of what you're going to have, or none of it potentially, if you're dealing with things that are really that threatening. But I hope no one else has to deal with it. I make a lot of information free on my YouTube channel, which is also called Zenthanol. And so if you want to check out some of that information, I'm just very passionate about people getting good info, kind of like what we did here in the presentation. And if you do need more detailed help, you can contact me at zenthanol.com on my website. And you can also, feel free to ask questions on my YouTube channel and also my Instagram at SyncAngel, which is a private Instagram account. I mean, it's public, but it's just mine, So I usually talk about things related to my profession as well. And so, yeah, a lot of times people do interact, it goes down in the DMs. I didn't plan for it to be this way, but that's the world we live in.
And that also goes for the rest of the members on this panel. I think that the crazy thing is how extraordinarily accessible people like Matthew Gates and Kevin from OK Calyx, Brian from Rubber Ducky. I try to make myself accessible as well. So, don't hesitate to reach out, and certainly don't hesitate to show some support to these fine gentlemen on this show. That's how we keep this show alive, is through our small businesses and through your patronage. Brian, what's going on or actually Kevin, let's go with you since you're in the middle of something there. Kevin, what's going on, brother?
Yes. Hey, Matt, thank you for being here again tonight. It's a fantastic talk. Had no idea that PM could drown. That was a good one.
Yeah, think about it.
That's really cool. That makes me want to incorporate, when it rains, with my IPM sprays and try to be as effective as possible. So really good information.
It makes me wonder if like foliage sprays are actually anti-PM just from that, factor. Yeah.
Yeah. Great information. This is one of those shows you got to go back and watch a couple of times. So thanks everybody for being here. Real quick though, I don't know if you can see that.
Oh, nice.
Oh, yeah.
It's a whole lot of bioactive fungus gnat eliminator I got from My Beneficials. So that stuff works, y'all. It is good. I transplanted to bigger pots. That's why I ordered some more, just to keep it on top of it. But you can go to my website, okcalyxshop.com. I've got my Azufuego. That's kind of a blueberry strain. Regs are for sale. I've got the straw jelly crossed to peach pistols that just got released. It's up. It's 10 films, $50. And then the IMO3, 10 pounds of IMO3 is going out for $85 right now. We've got a lot of really, really good looking IMO3 that I've made from Ozark materials, from materials in Tahlequah, and materials from my own land here in Bigsby. So got some good stuff. Go check out okcalyxshop.com. And as Mark said, man, our show is not sponsored or anything like that. So we just kind of do this on our own with our own finances, and we kind of rely on people to go help us out at our website. So we appreciate all the support that you guys give us by watching this and checking us out on other Instagrams and social medias. So Brian, how about you, brother?
Yeah, this weekend's gonna be fun actually. Saturday I'm going on a conspiracy show back with Mackie. We're gonna talk about I know a lot of things that are outside of cannabis. I enjoy cannabis, but that's not the only thing I want to talk about. So with Mackie and with Ben Bolin, who surprisingly I didn't know, is kind of a little celebrity in that world, man. I wouldn't even say the word little. That might be insulting. He's a big name in that world. Which was really cool to see because he seems into the conspiracy world, but not so fringy where it makes me cringe. Like I still like to back off. I mean, I'm not trying to be. I don't agree with everything that's out there. That's for sure. So I like being on that because I feel like it's a conspiracy show, but actually has a little bit of depth. What up, Chad? And then Sunday, I just actually this morning, I was talking to Kevin before I came on here. Matt Powers is doing his Our Future every January. He does that. I like that. Getting into understanding regenerative farming, entrepreneurship, all of the things that does make me get up and want to, attack the day in the morning. And Kevin's talk is on a whole nother level, to be honest. I don't think enough men are talking about the things that he's talking about. I don't want to give it away or anything. But at the same time, this is a free spot at Matt Powers' Our Future. So you're able to kind of go in there and see what Kevin was doing, and then mine will be released on Sunday as well. I'm talking about autophagy. I'm talking about ways that I was able to remove. I don't know if you guys can see, but I am slowly removing all of this dark purple that was underneath my eyes from just being a fool and eating sugar, usually every evening around this time. And focusing on minimizing my food consumption. Going the next level with fasting and autophagy, stuff that you've heard me talk about. I was amazed a few weeks ago how many people wanted to learn more about that. And so I think if you listen to Kevin's talk and you listen to my talk, it's going to be totally different than any regenerative farming thing that you would potentially hear. But in my opinion, to be an entrepreneur, it's all on you, baby. Nobody checks to make sure when we get into the office, if you will, to make sure that we're there. No one's checking to make sure that we're working later, into the evening. And so to be an entrepreneur, I think you have to focus on your health. Health is wealth. If you are run down, you're constantly drinking energy drinks and stuff, you need to focus on things like the adrenal glands, all of these things that I've taken for granted. I just share in a 1 hour presentation that took me about 4 days to put together. So if you guys knew how long that actually is in my world, it feels like 40 days it took me to put together. So I put it together, and I wanted to also shout out to Matthew Gates. I've been a part of interviews on him on a variety of different podcasts. I've seen him on a variety of different shows, learning from him. And the presentation, man, the work and effort that you put into these is next level. So I just wanted to highlight that. I mean, you were showing pictures and infographics and stuff that I've seen potentially in other books, but not in the visual sense. I mean, they might be bringing it up, but not in that deep sense. So what you had today and what you were showing is a very rare thing. I'm not sure you would potentially even see that in some of the, I think like Teeming with Nutrients would have been the first book that comes to mind. There's other books like it, but where he's speaking on this, and it's always kind of gone over my head a little bit in the earlier days. But to have this presentation, brother, was a eye-opening for, I think, the layman, the guy from Georgia that's trying to keep up.
I appreciate that. Mark, Brian, Kevin, the fact that you had some of the questions that you had, regarding some of these points, I think that it was really, really cool. For me, especially because it showed that people got what I was talking about, that you guys making extrapolations that I thought were very reasonable and were the reasons I bring it up. Sure, they're technical. Sure, sometimes, you have to like put me at 1.5 speed or something sometimes. But when you go and digest it and you come back and you go, it makes you go think, maybe I should try this. maybe I could try that. And that, like 100 people doing that sort of thing, that pioneering, like you said, very good word for it. People taking this cool new research and incorporating it in, and maybe they don't get the results they wanted the first time, but then they try it again, and more information comes out. So I'm happy people are even hungry for it, and yeah, I'm glad that you appreciate it. Thank you.
You're what I would call a diamond in the rough, because in a sea of just information that is it's a lot of times it's overwhelming because it's contradictory.
Yeah,
But you're a person that has probably the most integrity of anyone I've ever met in this industry. Who, without you're not we don't pay our guests to come on here. This is something that Matthew basically volunteered to do, out of the kindness of his heart, and to come on and to share actual real information, and to be willing to roll up your sleeves with us for 2 hours to nail this thing down is pretty amazing. And I think that deserves a huge applause, but definitely all the support in the world. And you can definitely know that by using somebody like Matthew Gates from Zenthanol Consulting, that you're going to get the actual information. You're not going to get rhetorical baloney coming out of a vacuum off of Reddit. And we've all been there before. We've all been so confused and so defeated from contradictory information coming all over the place. So the fact that you've got a source of information that is true, and if it's not scientifically backed, you're not willing to endorse it. And I really appreciate that about you, Matthew. So again, thank you for coming on. Real quick housekeeping, guys. Next week we've got Dragonfly Earth Medicine, just a small little name in regenerative agriculture, coming on. They're going to be sharing with us about how they close their loops. We want to make them accessible to you all so you can ask your questions. If you've got property, if you're thinking of property, permaculture, regenerative agriculture, metaphysical, these guys are in the thralls of it and have been living it for a long time. So super excited to talk with these guys. The week after, we have the man Josh from Yellow Skunk Farms, who's just completely blowing up right now with his what are we calling that thing, boys? The living soil.
The amusement park, baby. It's
The
amusement
regenerative,
park. Slip and slide. It's got it all, man.
We got a water slide, got plants.
The regenerative amusement park, baby. It's got the slip and slide. It's got the rock features. It's got the lazy river.
It's got the literal algae.
The lazy river. That's what it is.
Oh my God, it's so funny. But guys, we had him come on. It's got to be 4 months ago, 5 months ago now.
Nobody believed.
Nope. And it was so fun. It was such a fun conversation with him, because it was just like this dream session where we're like, oh man, what if you do this and that? And just, there was a lot of people that were questioning this system that he's building. If you don't know what we're talking about, do yourself a favor and give Yellow Skunk Farms a follow and check out this thing. It's incredible. And look at his plants. I mean, look how it's working. Look how it's actually flowing water. He's incorporated colored springtails, which from us. Woo. He's incorporating shrimp, fish. He's got natural moving, flowing water that he uses to water his plants. It's absolutely incredible. He's coming on, and he's got internet inside of the.
Yeah, it'll be live. It'll be a live dog and pony.
Yeah, so he's doing the dog and pony show and tell. It's going to be amazing to have him. And then the week following that, we've got Nerds Genetics coming on to talk with us about what they've got going on, and to hopefully answer some of your questions on breeding, so we can get further in on that side. You can support us by going to mibeneficials.com. You can support this show by subscribing, by liking, by sharing it with your friends. And also by supporting the guests that we have come on. It means a lot. And when there's a return on their time, it means that they're more willing to come back on our show. So that certainly helps out the show as well, by supporting our friends and friends of this show and community. Guys, hopefully, this was a heavy hitting conversation, especially compared to last week where we really slowed it down. And we really appreciate the 40 to 50, 60 of you that are tuning in live to this thing and keeping up with us. It's hard to keep a show tantalizing without jumping all over the place with some topics. And I think that every episode we've put together certainly has its application. And hopefully the information that we're sharing with you guys is something new that you can actually use in your gardens, something you can actually apply to your lives and to your hobbies and to your professions. Really appreciate everyone tuning in. Supporting us and supporting ourselves in this community is what it's all about, baby. So until next week, we hope y'all have a great week. Take care of yourselves. Good night.
Yeah. See you guys.